Modern hyperscale data centers are sprawling cathedrals of consumption. They pull megawatts of power from local grids and require literal rivers of chilled water just to prevent their processors from melting. Look closely at the biological processor inside your own skull. It executes continuous, high-level learning on about 20 watts of power, less electricity than it takes to illuminate a standard LED bulb. For decades, our strategy to increase computational power relied on Moore's law, named for Gordon Moore. The method was brute force: shrink the silicon transistors, pack more of them onto a single chip, and increase the clock speed. We have now engineered transistors down to the 2-nanometer scale. That is the width of just a few individual atoms. At this threshold, classical physics stops functioning, and the rules of the atomic world take over. When barriers between electrical channels become this thin, electrons leak across them via quantum tunneling. The result is massive, uncontrollable thermal waste. Training a single modern artificial intelligence model on this hardware consumes the electrical output of a small power plant. A human brain performs infinitely more adaptable cognition using only the chemical energy of an apple and a glass of water. Computing has hit a thermodynamic ceiling. To process information more efficiently, industry must look past engineered silicon and turn toward biological cultivation. In 2022, researchers at Cortical Labs in Melbourne successfully cultured 800,000 living human and mouse neural cells directly onto a microelectrode grid. They did not give these cells sensory organs or pre-written software. Instead, they took the vintage arcade game Pong and translated its mechanics entirely into pure electrical signals. Voltage pulses sent to the left side of the cellular layer indicated the position of the digital ball. Pulses on the right side delivered electrical feedback, signaling whether the paddle successfully hit or missed the target. Within 5 minutes of receiving these signals, the living cellular layer organized its responses, moving the paddle to accurately return the ball. The Pong experiment demonstrates that living tissue can interface with digital logic to execute goal-directed learning natively. While matching silicon's reliability and scale remains a generational challenge, the path toward a biological interface is open. Scaling this technology requires massive amounts of raw biological material. However, harvesting and using intact human brain tissue is ethically and logistically impossible. The solution comes from Dr. Shinya Yamanaka's Nobel Prize-winning work, which proved that cellular development operates on a reversible timeline. Introducing 4 specific proteins into a mature adult skin cell physically transforms it. This chemical process wipes the cell's genetic memory entirely clean. This creates an induced pluripotent stem cell, a blank biological slate capable of developing into any cell type in the human body. Standard Petri dishes cause these cells to grow into flat, 2-dimensional sheets. Flat architecture is insufficient for complex signal processing. Instead, researchers suspend these blank stem cells inside a 3-dimensional gel matrix, placing them into a spinning bioreactor, where they can exchange biochemical signals in every direction. The cells self-organize over 40 days, guided entirely by their DNA rather than an external blueprint. They form complex cortical neural structures. The bioreactor produces a highly dense, self-assembled 3-dimensional processing core, a biological substrate ready for digital integration, distinct from a conscious human brain. Connecting that biological core to standard electronics presents an immediate input-output bottleneck. Traditional silicon is rigid and dry. Forcing it into soft, wet, living tissue triggers inflammation, cellular scarring, and total signal loss. Bioengineers bypass this using microelectrode arrays coated in flexible polymers that mimic cellular membranes. Neurons open microscopic gates, letting ions rush across. The electrode pad captures this chemical shift, translating it into digital ones and zeros, establishing two-way communication. Neurons are not the only medium for biological calculation. At the molecular level, a single gram of synthetic DNA can store up to 217 petabytes of data, dwarfing the density of advanced solid-state drives. We are already building logic gates, and, or, and not switches out of genetic material and placing them inside living bacteria, turning single-celled organisms into mobile microscopic processing units. This integration has scaled up to fully remote infrastructure. In Switzerland, a company called FinalSpark operates a neuroplatform, the world's first biological cloud server. A developer anywhere on the planet can type a Python script, send it over the internet, and trigger microfluidic pumps and electrical stimuli in distant living organoids. Computation is no longer restricted to local hardware. The boundary has been breached, granting global access to biological coprocessors. Traditional digital logic is brittle. If a single transistor fails, the entire system crashes. Biological neural networks are chaotic and messy, yet remarkably resilient to noise and failure. To understand how these messy networks learn without software, neuroscientist Carl Friston proposed the free energy principle, also known as active inference. The theory states that all biological systems share a single driving imperative. They must minimize environmental unpredictability, surprise, and entropy to survive. When the cells in the dish brain experiment missed the digital pong ball, researchers fed them chaotic, erratic electrical stimulation. The cells experienced this as a high-entropy, unpredictable state. When they successfully hit the ball, the system rewarded them with clean, predictable, structured electrical signals. To avoid chaotic feedback, the cellular layer physically grew new dendritic branches, restructuring its internal computational pathways to ensure it received the predictable signal. The organoid did not understand the game. Biological intelligence is life's physical resistance against entropy. It rewires its own hardware to force order onto a chaotic environment. The immediate future of computing relies on a symbiotic hybrid. Traditional silicon manages digital transport and storage, while living organoids execute high-level pattern recognition and adaptive learning. In medicine, researchers are already growing living brain models from the skin cells of patients with Alzheimer's disease. This allows them to test millions of chemical compounds directly on living neural tissue, completely bypassing human trials. Scaling this up raises severe ethical questions. If a dense network of human cells adapts its physical structure to actively avoid chaotic stimuli, we must determine if it is experiencing a primitive form of synthetic sensation. The legal ambiguities are vast. If a biological processor is cultured from a specific donor's cells, it remains unclear who legally owns the living tissue calculating inside the server rack. Bioethicists are now racing to define the legal boundaries of organoid intelligence. We have to ensure that the pursuit of ultra-efficient computing does not inadvertently create a new era of biological servitude. Industry is transitioning from manufacturing to cultivation. For 3 centuries, our tools were defined by extraction and force, mined, smelted, and forged into rigid mechanisms. Now we are learning to grow and nurture technology from genetic code. The next generation of computers will consume nutrients, heal their own micro-injuries, and eventually age alongside us. We pushed physical engineering to its absolute limits, only to discover that life itself is the most elegant, resilient computational architecture in the universe. When we watch a network of cells learning in a dish, we have not engineered a new machine. We have built a mirror to our own intelligence, and the story of thought is only just beginning.